I'm Jenna Schardt. I work at the Allen Institute for Brain Science. I have been doing conjugate array tomography with Stephen Smith for the last 7 years. Prior to this, I was at FEI before they became Thermo Fisher, doing PLIM with the IPALM system that they had there, SEM, anti-EM. And I am now also part of the crazy people that Ian referenced who are doing full volume EM in cubic millimeter and beyond, which I think I also mentioned there. So I'm going to talk about how to really figure out your antibody situation for that conjugate or correlative, whichever word you like to use better, array tomography. So this is a section that of mouse tissue and primary visual cortex. We have all the layers. These are, we multiplexed. And so these are all of the antibodies that we used for that. We then took some conjugate bits, but I just figured this is a great overview shot to start with. And then as it zoomed in, we had a 3 nanometer per pixel resolution here for the EM, and our VOXA size for the wide field for the light microscopy is 100 and 3.8 nanometers per VOXA, like laterally. So we got pretty good resolution here. And yeah, so we all have been through now quite a few versions of what array tomography is. I'll just go quickly. We've been working in collaboration with Christina Machiva down at Stanford, doing a lot of just fluorescent array tomography. We've done some just EM array tomography, and then also, as this talk is about, combining both into one and figuring out really what works best for that. This is everything everyone has been talking about lots. So first, I just thought to set this up, I'd start going into our version at the Allen Institute of what we've been doing for tissue prep here. Sorry, this is a misleading title. I've got to change it. On the left is low accryl HM20, on the right is LR white. So you can see that there's a big difference in the amount of time that it takes, as well as what we do. We found, and this is our big one, is that we can just use regular EM fix and it still preserves most of the fluorescence abilities for us, even if we have native fluorescence in there. So that just really helped us preserve ultrastructure. And then we, for the low accryl side of things, learned that we needed to continue plunge freezing versus high pressure freezing. For about 5 years of this time, we were high pressure freezing all of our samples, which is on to the right. And you can just see that our ultrastructure was super degraded versus plunge freezing after cryoprotection, and we maintained quite decent ultrastructure there. Another thing, when we're doing our low accryl prep, we're using an AFS, an automatic freeze substitution. We figured out that we can get much bigger blocks of tissue if we add something that agitates inside of the chamber, kind of just giving it a little bit of kinetic motion there. So we bought these vibrating face washers, which our purchasing department thought were very strange, better than our alternative, which was going to an adult shop. We wouldn't need to get into that. But anyways, we put that in, we vibrate for about 5 minutes at the start of each resin or solution exchange, and it really helped us get to bigger blocks. We wanted bigger blocks because we are going for a pipeline of cubic millimeter samples and data sets, but if you need smaller, you don't need to do this, and everything works out. There's been a lot of talk so far about cover slips. We use carbon-coated cover slips up at the Allen. One, it allows us to kind of see our samples on here. We buy pre-silanized cover slips. If you have a large team and don't want to spend the money, you can silanize yourself. But we buy the pre-silanized cover slips. They're about $3 a pop from the Schott glass store. And we're using full-size cover slips for this. They have slightly cheaper ones, smaller, and then carbon coat them ourselves. So also have had multiple talks about ultramicrotomes. Because we were in such a large production mode here, we built ourselves a little robot that has a modified boat, which you'll see. So I'm just going to play this video here. So it goes over, picks up cover slip, drops it down, waits, we cut. So we'll just do a short ribbon here. You can see our knife was dirty, we had a bunch of knife marks, but it was just for the video to kind of see. Comes over with a whisker and picks this up. Those are piezo motors, you can hear them. So with that method, we are able to cut in 3 days' time a cubic millimeter of tissue. And that just allowed us to have a lot of reproducibility there. The only human interaction at the time was wiping off each ribbon and controlling it with a PlayStation controller. We've since introduced the idea of a quote unquote zero cut, which kind of sometimes separates it out off of that knife edge. Sometimes it doesn't work. We still have to get in there. And we've gone from doing about 25 sections per cover slip, which just seemed, we had these old chambers that we used to use, to doing anywhere from 75 to 100, depending on how tall or like in Y direction our sample was. Okay, so we're going to get in now to what my talk's supposed to be about, which is antibody testing and optimization. So we have, over the last 2.5 years, been working on a grant with Jim Trimmer, who ran NeuroMab out of UC Davis for a long time, of testing and optimizing antibodies for that grant. And so we've been doing a lot of this, but before that, we had to figure it out for these larger pipelines as well. So what we've been doing is taking a ribbon of decent size and then using a pat pin to separate it out. And in doing this, you get shorter ribbons, so you're not getting quite as many in a row. But really what we've been looking at is, can you track the same little dot of your antibody across 3 to 5 sections, depending on how thick I've cut it? So there's 2 things here. Of one, if you kind of have an idea or you're just doing a quick and dirty test, you can just put on 3 different antibodies into A, B, and C. Or you can do it if you have all of the same antibody, you do a dilution factor there. We've been holding one antibody constant throughout most of our testing, and it's one that we found very early on stained the synapse quite perfectly. So we've been using synapsin 1, which is a rabbit antibody for us, and that just allowed us to check to see if there's any co-localization there. This was because all of the NeuroMabs, being MABs or mouse antibodies, our other tried and true is a PSD95. Both of those, if you remember from that beginning image, we just really were able to line up those synapses quite well. So it's very easy to tell when an antibody doesn't work. So these are different clones here in green of pan-Maguk. So if it gives this background instantly, this was like a very, very easy no. I will say now, I am not a biologist. I'm not a microbiologist. My background is in chemistry and material science. So having a very quick way that I could just tell my boss that this was an instant no was great. When we got into samples that were more like this, which, you know, the green channel of the pan-Maguk, which is what we were testing, lights up, it's a lot harder for me to say on the fly if this was good. We have some colocalization here. It's kind of where it turns into that orangish yellow, but we are getting some of it into the nuclear, which is in, that's DAPI in blue there. So this is one that we went back and did some optimization in that concentration, and we found that there were a lot more of these colocalizations there. So these are the antibodies that I've been referencing, synapsin here. These are all of the ones that have already been tested for array tomography. This was as of 2019, so it's a while ago here. There's more data that's being published on this as the study wraps up, but a lot of this was done by Christina down at Stanford and some by our lab at the Allen Institute. So this is another sample from that grant that actually we said had some pretty good colocalization and that we gave the thumbs up. So this is synapsin 1, V-glute T2 is in green, which is pretty blobby, and I'll explain that in a second, and then DAPI. So this is registered in with multi-stack reg in Fiji. And as it's going pretty fast here, your eyes kind of see different things. But if you slow it down and zoom in, you can start seeing, yes, this has some chromatic aberration here. This was taken from the edge of the sample. I was trying to get rid of the green blobs for everyone's vision here. But as it goes a little slower, there is a little more of that like yellowish orange and not just red. And I know it's a little wavy. It's not my best work. But you can track the same blob kind of through and through and saying, oh, that is something that's actually real and not just hanging out for one section. So there's some that are big that are hanging out for one section. You can see if I have my mouse over it, it kind of comes and goes. But overall, there's a lot more here. So me working with said boss, Steven, was able to figure out, okay, this is real. We can give this, that this has positive, and we think that this could be a good candidate to make an actual purified antibody from a soup, which is what this is. This is from that testing. So all of this right here is because these are just big globules of primary and secondary that I did not rinse off as well. So this was part of a test that had, we did 300 plus antibodies. 150 of those were done in a week, which is quite a few antibodies to test in a week. So I believe this was kind of a final push for the grant a year ago. So yeah, so I mentioned in that last one, as the frames are going slowly, checking to see if your antibodies are somewhat in the same place, frame by frame. I put this frame slightly off. So over here, you can see we have this arch. That arch maintains. And you can see you have these two synapsins, which are over here, and these two synapsins. But all these little colocalizations here, which are very much in orange, and there's a lot more outside too, you can kind of just pick and choose, are still orange in the frame to frame, which is really how we kind of moved through everything. So yeah, because of that robot that I showed, as well as our wide field setup, we've been able to test close to 400, maybe a little over 400 antibodies. And we came up with our standard of what we use for a triple round staining. We produced multiple data sets of this, which we're still working on getting out into production and publications for everyone else to kind of hang out and look at. But these are our standard, as you can tell, we did a lot of synapse detection here. So mostly these all, and this is all, sorry, in mouse brain and human samples, typically from the primary visual cortex area in mouse. And then in the human samples that we got, we were just hoping that we would get something. The Allen Institute partners with multiple surgeons across the region. And if they're doing an epilepsy or Parkinson's surgery and the patient consents, we get a little bit of tissue. So overall, that's how we get images like this. And we can zoom in and really see how that correlates here. So you can see the myelin is quite easy. And this is actually for this, what we use to align. I've done both hand alignment as well as on our team. We had some very talented computational folks who wrote code to do this in mass, which is great because when you're doing a cubic millimeter and tracking EM2 is not the most fun. So yeah, TD tomato was signaling specific cells. That's why we have this RORB IRS Cree down here. It was specific to get sparse labeling so that we could trace full axons and neurons all the way through. And then synapsins in green. So if I zoom in a little more, you can see that that connects on the synapses there. And then GABA is in yellow. So these are all of our GABAergic cells, as well as Gefrin and all the other ones that you can read for yourself. So I can go through here and just have some pretty pictures for everyone to follow. So it looks quite nice. And then we can open up for a few questions. So yeah, this was, and these are all taken at 70 nanometer thickness through this. And we'll just go back so you can keep following your favorite dot. Ta-da. The one thing that we found, if you're going past just the standard 1 round here or EM, that multi-round staining, we get decent fluorescence to maintain all the way through, depending on what your original intensity is. And we think that's because the surface gets a little bit roughed up every time that we try to scrape those antibodies off. So after each round of staining, we would flush it with a high pH. We've since dropped this down to about 10, not 13. This is our image, our direct comparison of a virgin ribbon versus a flushed ribbon. Since we've dropped it down to that 10, we get that much better signal to noise there in the SEM. Yeah, and so we're able to check out different excitatory synapses here. Our research is mostly based in synapses, and this is just showing, this is on our most recent data set that we're working on getting a preprint out by the end of the year. I'm hoping that actually happens. As all of you know, in research land, it's not normal. So I'll follow. This is a big team effort. The Allen Institute prides itself on team science, and it is very real. So all of these people here are part of either the synapse biology team or the EM connectomics team. And we've all worked on various parts of this. And then, of course, Christina at Stanford, Richard Weinberg's out of North Carolina, and Nish does a bunch of our synapse detection. He no longer is in the EM world, but he still helps out quite often. And Eric Perlman has his own LLC, but works mostly out of Hopkins in Baltimore. So yeah. Any questions? I know I ran through that pretty fast.
Thank you, Jenna. Excellent work. Thank you for sharing with us. There are a few questions. I think there's one question in the chat, and somebody, Steph, is asking if you will share the information about the vibrating facial massager you have.
Yes. Yes. I can put a link in the chat. I'll find it on, it was like, you know, a $10 thing from Amazon. I'll find it. I think they like, it's like a knockoff of probably something that costs like $70 at Sephora or something ridiculous like that. But yes, I'll put it in the chat. It's
Great.
rechargeable, so it's very silly.
Okay. Melania, do you want to ask your questions?
Yes.
Sure. Yeah. Yeah, I'm just curious to know a little more about how you're doing the cryoprotection and plunge freezing on the big tissue pieces.
Totally, yeah. So there's 2 ways that I've done it. The cryoprotection is like a 2-day with an overnight step. We're starting off with sodium acetate as well as a lot of, oh no, I'm gonna get these 2 confused because it's still quite early here on the West Coast. It's either glycerol or glycine, and I can email you our full protocol there. Pretty much we're cryoprotecting with either of those, and then using Aklar spears, we you can either drop it in to the slightly frozen, for the AFS, we're using a 4% urinal acetate and acetone mixture, and I've dropped it into that with the Aklar spears to really good results. That's the one that I had. Let's bring that up. This one here is from that. So this is a spear that goes straight in, and that is at -90 degrees in the AFS. It's already in that. You can also do a very similar method of dropping it into methanol that's surrounded by liquid nitrogen, not straight into liquid nitrogen because it's too harsh. And those are 2 methods that we've used for that. And so I mentioned Richard Weinberg, his lab, and his lab tech, Chris, developed one of those methods, and then Forrest Coleman and Juan, oh shoot, I'm forgetting Juan's last name, at Stanford under Stephen Smith developed the one with the UA and acetone.
Yeah, that's cool. I would never have thought of that.
Yeah, so the UA acetone is my favorite because it's just a one step and it gets you straight into that first go that
Right.
you're about to do anyway, at least in our protocol. And so yeah, the protocols that we use, I can send out after this to Rue, and then
Okay,
I can distribute
Excellent.
that if wanted. We're eventually, like, they're not secrets or anything. We've just been working on them for years and have been quite slow to publish everything. But yeah, we're big on open science here, so I can definitely share and all of that also.
Thank you.
great.
Yeah.
Alice, you want to ask your questions?
Yes, nice talk, Jenna. And I have a question regarding of you do multiple round of immunolabeling, right? So when you finish one round of immunolabeling, and as I know that when you do before you do the rate tomography, you have to counter staining with heavy
Yes.
metal. But is that mask or the later of the epitope for the antibody binding for the second round, third round, how would that affect the antibody antigen binding?
So you're talking about when I mentioned like this step here or this one here?
No,
Okay.
after you do immunolabeling,
Oh, yes.
before you do the SEM imaging, right?
Yes.
Because it's HM20 processing and it doesn't contain much heavy metal in the sample, and you
Right.
have to do the post staining with UA and the lead, right?
Yes.
So I think the UA and the lead, maybe it will cover off the epitope of your sample when you do the antibody strip off. It will has affected those, with these two things together, maybe really affect the later multi-round of antibody labeling.
Yes. So we are only doing the UA lead, the heavy metal staining at the very end. So after all of the fluorescence, once we've done that, we have figured out we can't do any more fluorescence imaging. So you have to do all of your fluorescence first and then go through your heavy metal after that.
Yeah, that's good.
You can,
yeah.
We've stripped it a few times and gotten very faint fluorescence. Like it's usable in kind of like a Hail Mary situation. If you're like, oh God, I forgot the GABin, that's like very important for this. We've stripped it once or twice of the lead and the potassium permanganate that we use. The UA seems pretty okay since it's already been in there. And we've gotten some data, nothing that would be like a gold standard that you'd want to ever show anyone, but for your own like sanity's sake and a double check, you can totally do that. But that often just kind of looks like this little bit here. I know it's very small. If you see this tiny bit of glue in one, that's just like how the whole of everything would look. You're not going to get anything that has deep bright fluorescence there going on.
Thank you. That's great.
Jemima, you want to go ahead?
I have probably tons of questions, but maybe just one specific one that I would ask now. Have you ever tried your cryoprotection and plunge freezing with adherent cells on cover slips?
We have not at the Allens. We have only been using tissue for that. But our cell science group has tried something similar, and I don't have those results on hand or actually know if they worked. I know that they were trying to do it with like a slurry, I believe, with yeast in it, to kind of keep it all together, but I'm not 100% sure how that worked out. I can find out this afternoon.
Thanks.
Yeah.
Good talk.
Thank you. You too.
There's one question on the chat. Jillian, do you want to ask yourself, or can you hear me?
I see. How did the LR White samples compare to the low accrel samples? The LR White samples look absolutely stunning for fluorescence. The DAPI is better. The fluorescence is brighter. The signal to noise is great. For the EM side of things, all of the lovely membranes that you see here and the synapses are just completely blown out. and look atrocious. You can kind of sometimes see a dark line, which was probably a synapse. You can see dark circles, which were most likely these mitochondria. You can kind of see the outline of the nucleus, but anything more, there's no vesicles that you can see in the synapses. There's literally, like, you can't see the ER, you can't see any Golgi, like there's no other structure there. So it all looks like this would be brilliant in LR white, but we're not even getting that. There's no membranes going on that we can see. So somebody else has figured out a good way for that. And I think for us, that is because we're not doing any like plunge freezing or anything like that. We've tried the Kit McDonald method about 1,000 times and we've never gotten it to work. So we have tried LR white cold cured, we've tried it in heat. Neither of them are giving us results on EM. We've had a case of lab gremlins. We also haven't gotten our V-Glut T1 to work in the last year, and it's worked for 6 years. So maybe it's just lab gremlins. But our LR white looks really good for fluorescence and terrible for EM. So if anyone has tips or tricks or wants to chime in on things that have gotten that to work for them, please be my guest. I would love to hear it.
Okay, any other questions? Did I miss anything?
Can I ask one question, please?
Okay, Anasta, go ahead.
So you mentioned that you still put UA before the embedding, right?
Correct.
Okay.
For the low acryl.
For the low acryl. And why do you do that? Because it's washed out anyway. I mean, we wash it, kind of, it's a very awkward thing.
So it does preserve some staining, so you can see it maintain
That's what I want
bit.
to hear.
Yeah, so it has a little bit, and it's hard to see, but maybe I can zoom in. You can kind of see here, it has this hint of yellow tinge. And so we've done tests, and you can do a 0 post stain with this and get really, really faint in the SEM of like contrast there that could be useful for somebody. It does a little bit of fixation in that tissue processing that, like, I will call it osmium adjacent. So osmium is fixing it in a different chemical way, right? And really hardening that, and you're not getting any antigenicity there to be able to use those fluorophores and fluorescence and proteins. But the UA doesn't block those channels. It's not big enough, and it also just doesn't bind the same way. So it's giving you a little bit of fixation, a little bit of ultrastructure, and then just a hint more on the opposite side of before you do that post staining.
Yeah, I mean, I have a feeling that the UA messes up with the low acryl because it's the uranium is fluorescent, right, under UV.
So we're not seeing any background with it.
Okay, so you're using 1% UA, right?
We're using
No, not about background, but it's kind of, it interacts with the UV light and it starts kind of messing up probably.
Oh, got it.
Yeah, from the physics, I'm not really comfortable. So I decided to remove completely the UA. We had this conversation with Richard probably 10 years ago, and he said, put as little as possible UA because it messes up.
Yeah, so when we're doing this in the automatic freeze substitution chamber, we're finding that it's totally fine with the UA amount that we have in there. We've tried everything from 1 to 4% in acetone, but I don't think that it is completely necessary for it doesn't give you, the benefits do not outweigh the risk if you've had trouble in the past, is what I would say. We're very much a do whatever works best for you type of lab. So we've been fine with our UA.
Can we try without UA and to see the antigenicity, whether it improves the antigenicity? UA should decrease the antigenicity, no question, because it binds to the negative charges and stuff like that.
Correct. It wasn't appreciable for our large scale. It gave us the ultrastructure that it provided on the back end of the SEM imaging was enough, and we had enough signal to noise that we felt okay about keeping it around. Okay.
And after sectioning, you have a traces of UA, right? You don't need additional counter stain.
We
just straight from the kind of you know, if you put it on the SEM just right after sectioning, you still have a little trace of
You have a little
kind of
trace.
counter stain, right?
We do the heavy metal counter stain at the end, but you could just have it with the little bit of UA and you'll get some
Right.
contrast there. It's just not going to be that dark, deep one. It's not going to be anywhere
Yeah, because I mean, I've done just aqueous UA over 24 hours in EPOL embedding, you know, kind of just the very, very simple thing. And it gives you robust signal and block, and you know, in room temperature gives you robust, very robust signal with the FEI detectors and stuff like that. I use CBS. It's great, you know. But I'm a little paranoid. I have to try that. I have to try to push a little more UA.
And I will say, so we're using a Zeiss Gemini, which we love. It gives us really good results, but it is slightly the hardware of the Zeiss. And I might be biased because I came from FEI, although I've been on the Zeiss for longer than I worked for FEI by far. The Zeiss hardware 7 years ago was a little bit inferior to the FEI hardware in terms of the detectors, and they might have caught up now. I don't know. But that might be also where our slight differences are too. It's just our equipment, but it's working great for us and our purposes. So yeah.
And you're landing about 1 kilovolt, right? What is the voltage that you're using?
So my landing energy is anywhere from 1.5 to 2 kilovolts.
Okay.
Yeah. And I'm using a
because that was the problem with Zeiss. You know, they used to shoot about 5 kV to get the signal back to the.
Yeah. So my bias stage is at 5 kilovolts, and I'm like 7-ish kilovolts down through the
7 kilovolt with bias, right?
Yes. So giving me a 2 kV landing energy.
Okay.
Yeah.
Thanks.
And we use the 2 kV for faster imaging. If we can go a little slower, we do 1.5 to 1.75.
Yeah, because I don't use the bias. I just shoot very low energy electrons.
Yeah, we can't resolve the synapses without having the bias on for us. We don't get that clean, crisp image. So, but
okay. Thanks.
Yeah, thank you.